Hey everyone, we have a great discussion here planned. I just told Neil we're coming to him first, and he was a little surprised. So.
Okay.
Start thinking about the topic. We'll keep this a little light. I think if folks have questions, you should absolutely raise your hand or just interrupt. But ultimately, I think our goal here is to really spend some time dissecting this question of how a company or how those, in my role, think about investing in a given drug discovery, drug development program. You're going to hear from a couple different perspectives, different size of companies, different stage of companies. We really want to make sure that we're able to help the folks in the audience here understand what the considerations are, what the factors are, and I think ultimately, hopefully, as you take it back to your organizations, can make use of that information so you think about how to attract investment and attract companies into your spaces. So we're going to kick it off with a couple introductions just to make sure everyone knows where we're coming from, the perspective we're bringing. Then I'll come to you, Neil, with our first question. All right. So I was asked to moderate this largely because Nicole asked me to, and I just do whatever Nicole tells me to do. My name's Walt Kowtoniuk. I work at a place called Third Rock Ventures. We're an early stage venture capital firm. We work almost exclusively in biotech, some genomics and devices in there over time. I do most of my work thinking about how we can bring products to bear for patients with rare diseases. And I also chair Global Genes and RARE Act. It's been an exciting year as we brought those two organizations together. Of course, we'll have lots of time for Q&A. So Neil, why don't you go next and talk about yourself and BridgeBio?
Yeah, sure. I'll be brief. It's a privilege to be here. My name is Neil Kumar. I'm the CEO and founder of a company called BridgeBio, which operates in the genetic disease space, both monogenic disease and somatic cancers with clear genetic drivers. I've spent my career in the sort of company building or discovery science space within genetic disease. Had the pleasure of working with Walt prior to founding this company at Third Rock Ventures, and prior to that was in academia.
Thanks. Hi everyone, Steve Rodems. I am the Vice President of Research and Nonclinical Development at Travere Therapeutics. Been in the rare disease drug discovery and development space for about 25 years now, and had the pleasure of seeing a couple of successes make it through the clinic and into approval, and I've seen a whole lot of failures along those years as well. So that's taught me a lot about kind of what to look for in new programs, new assets, new drugs that come our way, and use those learnings now to help do that due diligence. Travere Therapeutics is a commercial and clinical stage biotech company focused solely in rare disease. Core therapeutic areas are rare kidney, and rare metabolic disorders. Recently got accelerated approval of a rare kidney disease drug called Filspari or sparsentan, and looking forward to starting a phase 3 study in a rare metabolic disease called homocystinuria with a drug called pegtibatinase. We are a virtual biotech, meaning we don't have our own labs, so we build our pipeline through in-licensing, acquisition, or partnerships, and both of those drugs I just mentioned were drugs that we in-licensed along the way. So looking forward to sharing some of my perspective and my learnings over the years with you today.
Hello everyone, pleasure to be here, it's an honor. My name is Ala Ahmed. I head the Global Medical Affairs team at Sanofi for rare blood disorders and rare diseases. Been with Sanofi for over 12 years. The medical affairs team covers the registries. Again, we have a Genzyme legacy of over 30 years in lysosomal storage disorders. We also oversee all the diagnostic services that the company provides for all of our rare portfolio. My own personal interest in rare has been, for over 20 years, and also one of my focus is patient-led evidence generation and also patient-reported outcomes. Been doing a lot of work in this space and interested to share with you what we've learned and also to participate in this conversation.
Excellent, thank you. So we're going to kick it off with the title question, how companies decide. Neil, we're going to come to you first and just give us a sense for how it is BridgeBio, today over time has thought about which diseases, which programs to bring into the company.
Yeah, I'll try to make it brief. I think the good news is it's not limited by therapeutic area, at least for us, and I think a lot of early stage opportunistic biotechs like us. It's not limited by modality. We have 3 or 4 different types of modalities, from gene therapy to small molecules, that we work on. And it's also not limited by population size. think we have one of the rarest rare disease medicines that are commercialized in molybdenum cofactor deficiency type A out there. We also work on patient populations and unmet needs that range into the hundreds of thousands of patients as well. So really, we have two things that we look for generally when we're looking at a new idea. The first is, at least the moniker is, the science is the science beautiful? And by that we mean, is the science of medicine actionable? And by that, in turn, we mean, because we're really discovery scientists at heart, how well understood is the function of the genetic mutation at the level of protein dysfunction, cellular pathomechanism, and then how that manifests in the tissue of interest and ultimately connects to symptomatology. And we can get into the details of that. There's some genotype, phenotype, but human clinical genetics provides, when well understood, a beautiful tapestry against which not only to understand what's driving a condition, but also how one might be able to best reverse course in the disease and target those conditions at their source. And then the second smaller piece of that is really understanding how one might be able to measure clinical efficacy and what the variance of that is over time. So that's the first piece, is the science beautiful. And then the second piece is the program NPV positive, which I learned at our last boot camp. A lot of people don't know, so I'll just say at a high level, NPV is the way that the people who are giving BridgeBio resources try to understand whether they should invest in Bridge versus something else over time at a certain degree of risk. And there are aspects of whether a program's NPV positive that have nothing to do with the job of anyone in here except for me, which is the reduction of cost of capital and making sure that my company is as efficient as possible over time so we can go into as small a patient population as we think where the science warrants. But there are things we can work together on in terms of driving that NPV. One is the probability of technical success. That gets back to the science. Two is the cost, and that is the stage of the program. For instance, I can work on very rare conditions, but typically there needs to be some starting point in the clinic to go after. Otherwise, if we're starting all the way at the beginning, 14 years away from product, it may need to be something that's a slightly larger unmet need. So those are the types of issues that we think about from an NPV standpoint, and we try to be very open in our discussions with counterparties on that. But those are the two things. Science beautiful, is it NPV positive? That's how we decide.
Steve, can you give us a perspective from a commercial stage company? Well, you're commercial as well, but commercial stage company, a little bit different stage of maturity.
Yeah. So I think there are a lot of similarities in what Neil said. I think the science, obviously, first. We, I mentioned our core therapeutic area, so we do have to be a little bit narrow, I think, in that aspect, as we are, looking in the kidney space. We've developed a kidney expertise over the last 8 or 9 years, and so we want to basically draft on that and use the expertise that we built with the KOLs, our infrastructure, and everything else to bring additional kidney therapies to patients. Now, it doesn't have to be that exact because we recently branched out and brought in a metabolic disorder. So I think, somewhat, I would say, agnostic or opportunistic, but somewhat within, some constraints there. So that's probably maybe a slight difference. We also have to be very, I would say, risk averse. I think it's a stage of our company. Most of our company is focusing on execution of the current programs, and so we don't have a lot of shots on goal to take on new opportunities. We have to make that one shot a really good shot. So I think we have to have very high bar, very high criteria to select that next indication. So I think that may be a little bit different in the stage of our company.
Risk averse, TA focus, that's what I heard. Two big considerations. How about Sanofi? How does Sanofi pick? Big global behemoth.
In a similar vein, so again, we start from the unmet needs in the rare disease space. So we already have a footprint in the rare disease space. So we start from unmet needs in adjacent diseases within the rare disease space, but also within the larger Sanofi, because you specifically mentioned a large pharma company. So all disease areas compete for the same dollars. So we have in specialty care, we have oncology, we have immunology, we have rare diseases, we have rare blood disorders. So in order to make the case for the development of a rare disease product, again, the unmet need has to be clear, the epidemiology has to be clear, the endpoints for the development program needs to be clear, and again, more predictability on the outcome, and that's being, decisions are made in comparison to other therapeutic areas as well. But given our history and our commitment to this space, that's always a starting point for the diseases that we look for in rare disease.
I heard in there a lot about the epidemiology, the unmet needs, and sort of predictable outcomes. How would you think about that then in terms of how would you describe Sanofi's risk tolerance as it relates to taking on a new project, a new rare disease program?
I wouldn't speak for the company overall, but I guess I think the more that the disease is well known, characterized, the more that the epidemiology is well known, the more that you create predictability. And we often discuss with the rare disease community about the role of patients in generating natural history data and helping also in de-risking the space by identifying endpoints that potentially you base the natural history data on, and that's also discussed with regulators. So when the company goes and have a conversation with the regulators on a specific design of a trial, there is an agreement on what is a proper endpoint for that disease. So the more that the disease is risk, the more that you're able to decide internally that, okay, this is acceptable in order to move forward.
Sure. And Steve, I heard you say that Travere is generally a bit, I think you said risk adverse. Neil, how does risk factor into the equation as you think about BridgeBio and maybe help calibrate a little versus some of the other organizations here on stage?
Yeah, I think for us, what it means is the bar is going to be really high on things that we look at. We really have to have things stack up. Stars have to align, I think, for that next thing that we bring in. And we're really bringing stuff in, I think, at the clinical stage at this point in our maturation of the company. So I would say that, you know, given some of the expense around the clinical stage, you can't take a lot of chances at that point. So, you know, we're going to look back into the preclinical research and really make sure that science is there and the translational work and everything else to give us, I guess, the best probability of success on a program. So, you know, versus I would say starting a bunch of preclinical programs that have various technologies around them that some may work, some may not. We're not quite in that mode to do those types of things.
Neil, anything to add?
Yeah, I mean, I guess we have a different take on risk, which generally is, if you sort of look backwards in time at the industry, it's really hard to figure out what drug's going to work and what drug's not going to work. Even for, I mean, we have Richard Scheller, Charlton, we have some legends in R&D in and around BridgeBio, but they still get it mostly wrong. It's a mode negative, expected return positive lottery ticket industry. So I think the way that we think about risk is knowing that we're mostly going to get it wrong, asking for a certain set of starting points that bias you toward right, which is basically the science that I talked about. If you understand mechanism of biology and you can target it at its source, that biases you to kind of a better parameter space within the overall risk conundrum, but you're still mostly going to get it wrong. So then we start with a lot of programs and they whittle down over time. And the hope is that you have failures happen early when you've spent less money. We are a discovery-based organization at start. You know, a lot of clinical trials and some commercial products now, but really that's where we get started with most programs. But we've had failures in the clinic too. So the way I think about risk is it seems like if you're going to be a biotech, you have to accept quite a bit of risk. And the way to finance risk and put a portfolio together in and around risk is basically to diversify and then bring investors in and around that. That's our theory of the case. It could be right or wrong. Yeah.
I like that. Don't know if you're right, but you know what you're going to go do.
Just, try not to be too wrong.
Try not to be wrong all the time.
Yeah, try not to be wrong.
Yeah, maybe, Walt, if I can just add to that, I think, you know, what we try to do is just try to stack the deck as much in our favor as we can as you're looking at the preclinical biology and, you know, looking for therapies that target root cause of disease and the genetics and try to get as many of those things lined up to give you the best chance of success. But you're right, there's no guarantee in drug development.
Yeah, so I mean, some of the things, I was taking some notes here, some of the things I heard, right? You said science is beautiful, but we heard, the science and really there being a mechanistic understanding, but that's consistent throughout what each of you have said. Steve, you really highlighted a little bit of a later stage, but Neil, I think you acknowledged that, later stage factors into the way you think about programs and new programs starts as well. And from Sanofi perspective, the one piece I heard that was maybe a little unique was you used epidemiology. Right, you mentioned that in Sanofi's thinking, epidemiology and presumably the prevalence incidence of the disease really factors in. Can you say a little more of that? Because I didn't really hear that from the other two gentlemen. How does Sanofi think about epidemiology and incidence around a given new program?
It is, I think we heard a little bit about the net present value of a certain, again, we are committed to rare diseases, but there is, I think, a threshold around epidemiology in terms of what would be a potential sustainable product. So that's kind of puts a factor on rare disease consideration. And again, as we move also into gene therapies, you mentioned prevalence and incidence. So the model also slightly shifts because now you kind of have a curve where you potentially cure, and then
Sure.
So epidemiology plays a factor on how we look at diseases.
There's a dream there, right? You convert incidence into prevalence.
Obviously, also there is a lot of uncertainty on epidemiology in newer conditions, and part of the disease awareness as a medical affair, and once the product is in development, the more we understand more about the epidemiology. So it's iterative, and you start with a lot of unknowns, and as you progress, you revisit your assumptions. So.
Steve or Neil, anything to add to that?
Yeah, I think I can add that. We're often asked, what is that cutoff? What is that threshold? And we don't have an answer to that because it depends. There's other factors, the cost of the program, all kinds of stuff that kind of goes into that. But also maybe creative models of how to do this for smaller diseases, risk-sharing models. We have a discovery collaboration with NCATS that is really a risk-sharing model around a rare disease. And I think trying to think about, I'd encourage the whole industry to try to think about what are ways that we can start ratcheting down that number and treat smaller and smaller diseases. I've seen that number come down over my career. My previous company thought cystic fibrosis was too small at one point, and is now the basis of an $80 billion market cap. So I think creative ways to either finance or do those drug discovery programs, I think, is needed.
Yeah, I guess I would just add, just from office hours earlier today, there's a lot of technology out there now, a lot of databases that I think people are just starting to find out about, certainly at this conference, that can really help revise your understanding of incidence or prevalence. And I'd encourage everyone in this room to get familiar with some of those tools, not just from the standpoint of sort of reducing the error bars around the specific condition of interest, but also connecting that condition of interest, and I know we've talked about this in the past, to other conditions. There was a seminal paper that just dropped yesterday, I think, on alpha missense out of the deep fold group, where you can actually use these technologies now in the context of the gene of interest and connect it to a whole host of more broadly described traits and specifically larger diseases. And so I think when we talk about epidemiology, we're talking about a condition, but there's also the related conditions if you've created a tool to go after one condition. So everyone should be thinking about that always. I think it's a really interesting part of what we do.
It's a great segue into the next question for this group, which is really trying to flip it around and really what can we do to encourage the folks in this room as advocates to start building and creating within their community. We've talked about some of these factors, but how do we flip it around now? What advice do we have in terms of how folks in this room can make the disease or the patient groups the patients they represent more attractive for pharma biotech investment? Steve, coming to you first.
So I think there's been a lot of sessions at this conference about research readiness and other types of things. All that stuff is important. I think the more you can learn about your disease, learn about the mechanisms, and present those to companies and present a good data package, I think the better the case. I think obviously having a strong patient foundation is something that we look at, something I look at personally. I think having patients involved early on is important. And so if we're evaluating a disease, I want to know what the patient foundation is for those diseases, because that helps a lot, helps us a lot. We need to learn from you guys. You guys are the experts in your own disease, more so than some of the researchers and some of the doctors. So we need that information coming from you.
What would you add?
Yeah, I think from the patient side, we just discussed discovery, science, and credentialing, moving into development, regulatory conversations, eventually labeling, and then there is another barrier, which is health technology assessment, et cetera. So across this journey, there is a role for a patient organization and for the patient. One area is, the unmet need, because with every stakeholder we interact with, trying to explain the unmet need in a specific disease is you start from scratch. So whether it's a payer or a regulator or any other stakeholder. So I think one of the biggest role is to make the unmet need in the specific disease very clear. Also translate that qualitative data into also something that is more measurable and quantifiable. So any decision maker can be able to make evidence-based decision making. So I think that's one piece. I think we discussed the role of natural history, which also helps a lot in trial design and in conversations with regulators about comparator, what's the effect of the drug, because you could have a scientific underpinning, you could have a biomarker, but what does this decrease in a biomarker, what does it mean for a patient? That's something that patient groups can help with a lot. From the industry side, and back to the innovation, because we could innovate with regard to small sample size with the basket trial, which is you pick a disease with an epidemiology that is more common, but you also, on the same pathway, there are diseases that are less common, and you try to piggyback them in the same study. So that's one way to study multiple diseases. With regard to fail fast, there is also the concept of umbrella trials, which is you try different mechanisms in the same study and see which mechanism works. So kind of you fail all the others and try. So there is innovation also that's coming from the company side to address that, small amount of patients really trying to get to credentialing fast and also to address the unmet need.
How about you, Neil? Anything to add? I mean, we had some really good suggestions in there.
Yeah, that's pretty good list.
I think there's for me, one super simple way I think about it is, folks in this room are often representing a given patient community and really understand that disease super well. Folks on the stage here are often looking across multiple different potential drug programs, multiple different communities. There's a huge piece of just sort of helping sort it out and figure out what's real and what's not. Beautiful science, right? Who are the right investigators? What are the studies we should be looking at? How do the pieces fit together? You mentioned natural histories, right? Do your RARE-X. If you're not in RARE-X, go talk to Nicole about getting in RARE-X. But how do you really make sure the phenotype is characterized over time so that folks who are looking more across the horizon can get deep quickly? I don't know if anyone here would second that or modify what I said. No takers.
No taker. Well, I think I think what you said is right. Is I think the natural history studies are very important. The RARE-X work is very important. Even the real world evidence that you can gather. I know we had to do a lot of this for our work in homocystinuria because we are looking at a biomarker, so we had to know how that biomarker relates to disease phenotypes. So that comes from a lot of that historical work, whether it's a controlled natural history study or real world evidence.
So I'll keep asking questions all day, but if folks in the audience have questions, please throw the hand up. Oh, there we go. I can't see your name tag, I'm sorry.
Does anybody else know my name already? I love to ask questions. So my name's Kevin Fried. I run a small company called Salem Oaks. We try to work with these foundations and everybody to educate them about this whole process. And my question, my comment first is, these people are very passionate about what they're doing. They're investing their own tissues, their kids' lives, all of that. And they need to decide which of you to work with. So they're doing the same calculus you are in the other direction. I want to know if you guys have any initiatives or programs for brokering the deals so that a foundation can actually work their way toward this golden thing that you have, which is epi. You've got endpoints that are not defined for some of these things. You've got enough of a threshold to meet. Do you have any programs that sort of broker that deal and help coach them toward you?
It's a great comment. Yeah, I mean, it's something that I've been talking about with Nicole for a long time, and I don't know if Yale's around here these days, but oh yeah, there she is. I mean, fundamentally, there's like a 25-slide loop that I think about every time I'm a scientific advisor for a new parent or a group of people affected by a condition. And we could do a better job of just throwing that up. But we've talked about this concept of a team room in a box and funding it. So how do we help for the next new idea, say what to do, and I actually think equally as importantly, what not to do. I don't need 30 animal models. I have human clinical genetics. I don't need, iPSC cell lines, like, all of them. Like we could just, and so I guess the short answer is I don't have that yet. I'm not sure that, should Bridge do that? We've been talking about a Bridge not-for-profit too, because there are also a lot of programs now that don't, unfortunately, they're not for the for-profit world anymore. The for-profit world is sort of tough these days in biotech, and so,
It's a kind way of saying it.
Yeah, and so, but this is work that we should and have to do. So how do we organize it, and then who should be putting forth that team room in a box? I guess it should be us, honestly. I mean, we haven't done it yet, though. It should be us. Maybe it should be Global Genes, too. I don't know. Definitely, it should exist, though.
But Sanofi has one set of criteria. Travere has another set of criteria. If you want to attract these foundations to come work with you, they need to know your specific criteria. What's important to your company?
Yeah,
And
definitely.
so how do you coach them that
so
closely?
Fine, you can set up the company. I don't see it as a deal and broker. I think all that language is bullshit. Like we're trying to do a really hard thing here, which is to take things that don't necessarily fit this super for-profit world and do the right thing for humanity. So I don't like if it goes to Travere, that's great. If it goes to Sanofi, that's great. I think we've got
No shortage of diseases to work on.
8,000 monogenic diseases that are begging for some sort of an initiative, so I'm not worried about that. But yes, I agree, every company can have its own set of criteria up, and we have a website splash page for that. We could probably do a better job of articulating it.
I do think there's a huge piece of this that it's still a human business. So anyone in this room, get to know the people you're going to partner with. That's true in anything you do, not only potentially working with a biotech or pharma, but it's still very much a people business. Figure out the mission, the values of the organization, what really motivates them, why are they staying late into the evening to work on something. None of that's actually what we talked about, by the way.
Yeah, so we don't have a program, so to speak, but we've had a situation. This is a small community, right? So we've had a situation where we had a collaboration with NCATS around a disease called NGLA1 deficiency, and Chris Austin, the former director of NCATS, was talking about that. And someone came right up after that session and said, hey, we want to do the same thing with our disease, Alagille syndrome. And so that's how a program got started. So obviously I think in this small community, word of mouth and getting out there and talking about things.
Maybe I would add, I don't think that the patient group should work based on a company criteria. I think the disease should have a criteria, and that's agnostic to any company. So there are diseases that are mobility disorders, that are speech disorders, and you could potentially standardize how you make assessments for every organ system in the body, and potentially agree with regulators on what this assessment look like. Because again, natural history is an investment from the patient community, so you want to collect the right data that would be helpful for any company to make conversation. I think where the company's criteria become is the technology platform, because if the company has a specific technology platform, it can only address X and not Y. So that kind of would drive, okay, it would work with this disease, but not with this disease. But the diseases themselves, any company should approach the same disease, and it should be what we're saying, de-risked and demystified, that regulators will agree on this endpoint because there is a conversation that maybe even happened between the patient group and the regulator before initiating a natural history study. So you know that what you're collecting is valuable from a regulator perspective. But that's clear.
My name's Tina Irv. I work at NCATS in the Division of Rare Disease Research Innovation. The group I work with is the RDCRA and the Rare Disease Clinical Research Network, and our primary focus with these grants that are funded by 10 different NIH institutes is clinical trial readiness. The question I get from the investigators that are part of this network and from the patient advocacy groups that are partners with these groups is how do we get the attention of industry? And the answer that they tell me that they most often get is, don't call us, we'll call you. We'll let you know when you're ready. But is that the best model? And what should I tell my investigators and my patient advocacy groups to do? Because they're lining up, they're doing exactly what you all said they should be doing. So how can we get your attention?
Well, definitely don't wait for a company to come to you. I think that probably everyone on this stage has a group whose sole focus is to go out and learn more about exactly and precisely efforts that you're talking about. And I hope that they're fairly easy to identify for BridgeBio. And we try to place them everywhere, at every conference, at every TLO associated thing, IP conferences, et cetera, and including conferences like these. So I would definitely be proactive in reaching out. We probably have at least 600-700 conversations like that every year. So and we welcome them. It's a big part of what we do.
Hi. Jay Vivian, I'm a researcher at Children's Mercy in Kansas City. I want to know what your company's interest is on preclinical data of repurposed compounds. Is this something you ever approach, or how's an academician with some preclinical data but on an FDA-approved compound of any interest to your companies?
Yeah, I mean, I can start. I think, again, it kind of goes toward mechanism and understanding the disease biology. And if that repurposed compound happens to fit very well with a mechanism in that disease, then yeah, I think that makes sense. I think, there are some challenges, I think, with repurposed compounds that may, be more symptomatic, that treat a certain symptom, but maybe not a root underlying cause of disease. And if you're looking at the wrong endpoint in the clinical trial, or you don't understand how that might translate into a clinical benefit in a trial, then that's a tougher sell. But I think repurposing is obviously important in this community, but it does come down to how it fits the biology for that disease.
Hi, this is Harsha from Indo-US Organization for Rare Disease. Great discussion. I had a question around diseases with varying prevalence in different parts of the world and among different subpopulations. So when we talk about epidemiology, prevalence, incidence, how do you take certain diseases which may be disproportionately affecting, say, South Asians or Africans and other communities when your early R&D and also eventual global launch, right? Launching in US, Europe, Japan, that's well-known pathway, but are there any strategies being discussed to launch in other countries, and how to engage these other populations in this rare disease research and revolution?
Talk about it from a Sanofi perspective.
So again, in rare diseases, we do have a humanitarian program that's over 30 years old, and it's something that we're proud of. And it was imagined at the first launch in the US for Ceridase, the first product in Gaucher disease. The humanitarian program started immediately in parallel. And recently we have a product in ASMD, or acid sphingomyelinase deficiency, and we launched the humanitarian. It's a very costly product in terms of cost of goods, et cetera, but the humanitarian program immediately was launched in parallel. And we have patients from many places around the world where we don't expect to commercialize. So the idea of access, global access, is always embedded in the launch, and we do it in parallel. We don't sequence. Once you have a label, you launch your humanitarian, because it has to be on label.
Yeah, maybe to pick up on the research part of that, I think we can do a better job, and this is something we've only recently started doing at Bridge, at identifying, to your point, founder mutations and populations, and then trying to go out and establish clinical trial sites there. So in the context of organic acidemias, we just kicked off a partnership in the Middle East where there is a founder mutation, and likewise for ATTR cardiomyopathy, we're just opening up our first trial site in Africa because there's an Afro-Caribbean component to the hereditary form of that disease. We'd done it once before in Tenerife with primary hypersoluria type 1, which may be a disease you guys have looked at too, where there's a founder mutation, but we could do much more of that. I think that's important.
Hello. I'm Jeff Kaufman. I'm with the Adenoid Cystic Carcinoma Research Foundation. I have a couple of related questions. So when you're looking at a new indication, you're thinking about developing a therapy for that. How important is it to think about whether there are second, third, fourth indications that would be appropriate for that in your coming up with the net present value and so forth? How simple is it to do that? How frequently do you go about doing that? And sort of a related question then would be the Inflation Reduction Act and sort of the not getting the market exclusivity for second, third, fourth indications. Is that working into your thought process of how you would develop a drug?
Yeah, I mean, maybe I can address that one because we see this a lot in some of the kidney diseases too. Is multiple potential indications, based on mechanism, and yeah, you do try to think about where else could this the drug work, and so that's an aspect of the equation for sure. I think we've got to be a little careful because sometimes it can seem forced. It could seem like companies that come to us and say, "Look, I have a rare disease asset, but I can also use it in this larger indication," and it's maybe the genetics is there or something. But those smaller companies are trying to increase the value of their asset, but it may not make sense really in that disease. So you kind of have to parse those out a little bit. But for sure, if you had a bit of a pipeline in a pill type of thing, that's an advantage to a lot of companies.
Yeah, I haven't faced the IRA issue yet, although with our ATTR asset, it is a small molecule Med-ED therapy. So if it had obvious other indications to go after, I suppose we'd have to think about it. It seems sad, and I hope that changes. I think it will. Actually, I just saw last week that people have taken to modifying that aspect of it because it really does, or will put a hamper on Med-ED drugs, of which there aren't that many, to be honest. And most people are working on antibodies or gene therapies or whatnot. So. But I think on the second, third, fourth indication, oftentimes it can be very obvious just based on what else is happening within that gene, and we tend to miss it. Like we have an achondroplasia program that also could be really relevant to a condition that's very similar called hypochondroplasia. It arises from gain of functions in the same gene. We overlooked that when we started the program, but now we're heavily involved in both areas. So. That's something that we should have taken into account earlier, and I think could be profound both for the folks you're trying to serve as well as for the company that's developing the product.
I think we have a couple. I was going to ask you want to go ahead?
Sure. I'm just curious. You mentioned being risk averse, which I think you're in the wrong business, but I'm wondering, given a lot of companies that have run out of runway, can't raise money, have programs that have been shelved, or do you view those as being de-risked in any way, or are they opportunities for companies that do have resources to develop?
Yeah, I think that's I mean, it is a tough one. I mean, money's tight right now, right? And so you could think of people running out of money on good programs, and I'm sure that can happen. I think also one thing that we've noticed in our due diligence is we get presented with a lot of things. Only a handful of those things I think are really truly promising, and they're programs that have struggled along the way, and that's why someone's trying to offload them, or they're running out of money because other investors don't have the confidence in that drug to continue to fund it. So if a company's struggling, they're going to all the other clinical development companies and saying, here, look at, can you invest in my drug? And so a lot of times stuff that's available out there, quite frankly, isn't really all that good. So so it's a challenge to say, okay, which ones are kind of failing because that company is truly running out of money versus which ones just don't have the right data around it to give you confidence.
Maybe just to add on, I do think that there are a lot of de-risked, credible starting points out there, given the capital markets environment, that could be interesting to a company. But I think more so it would be interesting to this idea of a not-for-profit that I was talking about earlier, that I think I've spoken to at least 10 people about today. I see Craig over there. Because I think a lot of the reasons that those programs, unfortunately, are not attracting capital is that the bar for capital today is very different in a high interest rate environment, in a completely different marketplace than it was 3 or 4 years ago. And so we need to find a different way to take those de-risk programs that are de-risked from a scientific and clinical standpoint, but may not be commercially de-risked, and move those forward. I think it really is the challenge, one of the great challenges for those of us that are trying to build entities to move important medicines forward that we need to address. So I don't know that a company should be buying those. I think a not-for-profit could be moving them forward.
I just say thank you to the group. It's a thorny topic, and I think we all probably get asked it in different ways, different times from various patient communities. It's great to explore it a bit here for 45 minutes with each of you. Thank you. And thank you to the audience.